All right, so this is a bit of a little guided practice for doing these Punnett squares. So here I am on slide 46 that you guys will be seeing on your own, but it's asking us to perform some Punnett squares, a genetic cross. And we are trying to cross a heterozygous parent with another heterozygous parent, and the allele that we're using is the letter B. Alleles can be any letter of the alphabet. Sometimes you guys are going to have to choose your own alleles and come up with these combinations, but we have our allele right now. It's B, so we can have dominant B or recessive B, and both parents are heterozygous. So the first thing you do when you cross something is you want to make a cross. A cross is a box with 4 spaces. And our heterozygous parents, remember if you go back to your vocabulary, is talking about dominant with recessive. It's when the alleles are not the same, hence the term heterozygous. And so we're using the letter B. So one parent is going to be dominant B, recessive B, because it is heterozygous for this allele. The other parent is also heterozygous, so we have the same alleles here. Dominant B, recessive B, both parents are heterozygous, and now we need to cross these. And so one parent I'm going to place up here, pardon the handwriting. The other parent we are going to put over here, and that is us ready for our cross. So when we make a cross, these alleles are coming from the top down and from the side over. And so what you want to do is pretend you have a person in the box grabbing for these alleles, because ultimately what you're predicting are the offspring possibilities for this allele. So if I have like a little person, let me see if I can draw a little person. I got a little person in here. This is the future offspring. The offspring needs 2 alleles, all right? It's going to grab one from the top, and it's going to grab one other side. It's going to grab one from that other side. So if, I just want to see if I can do this with the video. If I am in this box, if I'm looking for alleles, I'm going to grab this recessive B, and then I'm going to grab that dominant B, and I'm going to bring them into this Punnett square, and that's how Punnett squares work. Dominants go first. Same thing for every combination. All right, and so for these boxes, I've got these possible combinations. Let me see if I can change color real quick. All right, out of the 4 out of 4 would be 100%. One of those 4, 25%, have a possibility of coming out homozygous dominant. That means both are dominant. 50% chance of us producing heterozygous offspring. Heterozygous meaning we have dominant and recessive together. And then another 25% chance of creating a homozygous recessive offspring where both are recessive. Now, depending on what these things mean, this could be a different, that's the genotypic frequency. Depending on what those alleles mean, dominant genotypes where we have the dominant allele present, that could mean that we have a 75% chance of a dominant phenotype, and that could maybe mean we have a 25% chance of a recessive phenotype. Remember, genotype is just what we have these allele combinations. Homozygous dominant is a genotype. Homozygous recessive, heterozygous, those are genetic combinations. Those are genotypic combinations. But phenotype is what something physically or characteristically looks like. It's the traits that get produced. So 2 different things there. All right. That is your bit of a guided intro into Punnett squares. But now I want you guys to try it out.